Everyone who's tuning in. We'll wait a minute or two for everyone to try to arrive before we get started here, but welcome. Okay, I think we'll go ahead and get started here. Good morning and good afternoon, everyone. Thank you so much for joining us here at the webinar on computational approaches to protein condensates. And we'll get started with the webinar in a second. And I want to just make a few Protein Society announcements here. I'm Chuck Sanders. I'm the president of the Protein Society. And we're very excited about our upcoming symposium this summer in Boston, July 13th through 16th. Mary Munson and Ruben Gonzalez are the co-chairs, and they have a wonderful chair committee that is working with them on the program. And we're really excited about this. Registration opens in February 2023. If you were at the San Francisco meeting last year, you know it was a really wonderful meeting. And we're hoping for a meeting of equal quality or better again this year in Boston. So we hope you can join us. So I also want to just point out that, you know, the Protein Society sponsors a journal, Protein Science. The editor-in-chief is John Curian. Coincidentally, he's my new dean here at Vanderbilt. And we have senior editors Nir Bintal and Aitzabir Carter-Harina. And we hope that you'll consider sending your best stuff to Protein Science to publish. So if you don't read our newsletter, I want to call your attention to an article that came out just a couple of days from Dr. Vanita Sood. Careful protein scientists make excellent drug hunters. Oops, I'm sorry. This is a really excellent article, and I would encourage you to read it. If you don't get the newsletter, you can access it at the journal website, which is listed here. Very simple website. Or if you want to subscribe to it, just shoot an email to our staff. And so we hope you'll do that. And so let's get back to the webinar. Again, we're really excited about this webinar. And the organizer today is Monika Fuxreiter from the University of Padova. And so Monika did her PhD work in Budapest and came over to the States and did her postdoctoral work here in the States, and then went back to Budapest, where she was at the Institute of Enzymology for a number of years. She did visiting professorships at the MRC and at the Weizmann Institute. And she is an expert on how fuzzy complexes work and their roles in higher order protein organization. And in fact, she's the person who coined the term fuzzy complexes, which is now, I think, a pretty ubiquitously used term. In 2020, she moved to the University of Padua, where she is a full professor. And so I'm going to turn the floor over to her, and she will introduce our panelists and get the webinar started. Thank you, Monika.
Okay, thank you so much, Frank. So welcome everyone to this webinar. And protein phase separation is actually an emerging yet controversial field. I mean, clearly protein condensates contribute to cellular functions very importantly, but yet how and why they are formed, the solid physics foundations are still lacking. So today we will look at this phenomenon through the eyes of molecular biophysicists and how computer simulation and other kinds of computational approaches gives an insight about this exciting area. So in my talk, I will give some general introduction, and in particular, I will address the interactions that are forming condensates and also their cellular context dependence. So proteins form communities in the cell that represent many different levels of higher order organizations. Now, these higher order structures range from solid-like states, like the amyloids, to liquid RNP granules, including many different kinds of signaling complexes. But the common feature of all of these that function is that we're determined by a collective behavior of the molecules. This is in contrast to what we have in laboratory, when protein's activity is largely independent of external factors. And this actually encourages us to put forward deterministic relationships between sequence and functions of proteins, which is mostly dependent on the intrinsic properties of the protein and largely independent of the context. But it's quite challenging to apply this relationship to explain cellular functions, because their protein activity is not at all independent of other molecules. There are myriads of stochastic signals, and the noise level is high. One of the consequences of this is that the most stable state of all proteins is a solid-like amyloid state. So if we think about cellular functions, in addition to protein structure, we also need to take the concentration into account. And we need to realize that at high concentrations, such as in the cell, the condensates, the solid-like amyloid state in particular, but also liquid-like droplet state, are more favored in comparison to the native state. Although the specific details of this phase diagram are yet to be elaborated, it's becoming increasingly clear that perturbations of this fine-tuned equilibrium between the three fundamental states is causing protein dysfunction and a wide range of human diseases. So the question is, how protein sequences encode this fine-tuned equilibrium between the three fundamental states, and how do they encode the condensates, the droplet state, and the amyloid state? And to start thinking about this, we need to consider that this is a universal phenomenon, and the signatures must be present in the sequences of all proteins, or of most proteins. The other thing that we need to keep in mind about this, that formation of these condensates depend on the cellular conditions. Therefore, if we put some kind of interaction motifs into this holy grail, they may escape under different cellular conditions. So therefore, we may not be able to establish magic motifs, and we may not be able to fill this holy grail with specific interaction sites. So let's think about it in a very simple manner. Let's analyze the interactions. In the solution, the energy of the system can be obtained as a sum of the potential energies of the particles. If you concentrate, then the energy of the system will be the sum of the pairwise energies. But if you move to a dense system, then these pairwise energies will depend on the presence of other particles. And these many-body effects make the calculation pretty challenging. So this is a similar situation in protein sequences. Protein sequences encode many different kinds of motifs that cannot be simultaneously optimized. So therefore, they present some kind of perturbation for each other, generating ruggedness of the energy landscape. Well, this is a well-known framework for protein folding and have been used for decades, but this is very analogous what's happening with protein interactions. If we have many different kinds of motifs that act, let's say, under different cellular conditions, they will generate some kind of perturbations for each other. and essentially lead to not a well-defined, but a rather heterogeneous bound state. Let me illustrate this. We like to see this situation when proteins adopt structures upon binding. But if we analyze the energetics of this, we find a high density of suboptimal contacts. And therefore, this energetic frustration may lead to multiple bound configurations simply to optimize the system. So what we need to realize that similarly to protein structures, also protein assemblies may range from ordered to disordered states. And it does not necessarily depend on the structuredness of the protein. Both ordered or disordered proteins can form assemblies with well-defined contact patterns. This is an ordered binding mode, or can form assemblies via heterogeneous contact pattern. This is a disordered binding mode. And again, irrespective whether the protein is structured or disordered, most proteins can sample actually both binding modes, depending on the conditions. So the question is how this is encoded in the sequence. Well, these binding modes are encoded through a local sequence bytes, which essentially expresses the difference between a putative binding site and its environment, its flanking sequences. The difference can be computed using an abstract set of properties, okay, can be physical chemical properties. It's basically the difference between the information contained in the putative binding site versus the flanking sequences. If this putative binding site is very different from the flanking sequences, then the bias is high, so we can find a few distinguished residues that will establish, again, well-defined specific interaction. Bias is low. That means that the putative binding site is fairly similar to its flanking sequences. So you will have lots of ambiguities, and essentially you generate a disordered binding mode. However, these putative binding sites are not known a priori. And also their presence depends on the cellular conditions. So we need to understand that they may change, and we need somehow to estimate these changes. Let me illustrate using the example of TDP-43. So these are three different regions of TDP-43, and we computed the probability of the binding modes under different conditions. You can see that the amyloid core of TDP-43 may sample both ordered and disordered interactions at a really wide distribution. In contrast, the residues that are very far from the amyloid core and most likely promote droplet formation, they have a very narrow distribution. So based on the width, based on this distribution, one can estimate the so-called multiplicity of binding modes, which is essentially a likelihood that the protein region will change its binding mode if the context changes. So let's practice, let's apply these principles. If we want to compute whether a protein region is capable to be engaged partitioning condensates, we need to compute the probability of disordered interactions. These are essentially the medians of the distribution I had just shown so far. You see the example of alpha-synuclein, and one can observe that at the C-terminal region, the probability of disordered interaction is pretty high. So this is the region that is going to promote droplet formation. If we want, however, to estimate whether a protein is capable to undergo spontaneously phase separation, then we also need to consider that these disordered interactions need to be stabilized, and they need to be stabilized similarly to protein folding via hydrophobic effect. So if you compare alpha-synuclein, which is a hydrophobic patch at the NAC region, it more readily phase separates in comparison to beta-synuclein, which lacks this hydrophobic patch. So if we apply this prediction methodology to the human proteome, we find that about 40% of human proteins are capable of spontaneously forming droplets. In addition, 25% of human proteins can partition into droplets through droplet-promoting regions. So this is essentially demonstrating that without assuming any specific interaction sites of any specific chemical nature, one can account for the universality of droplet formation. And now let's try to see what's happening with the droplets when conditions change. Okay, today I will address this problem of droplet maturation and in particular aggregation into fibrillar structures. So what we need to realize here, that while the droplet state is stabilized by disordered interaction, the amyloid state is stabilized by ordered interactions. Both are non-native. So when the droplets maturate, basically they shift the binding modes from disordered to ordered binding modes. Now this is a process that cannot be captured in one dimension. We need some kind of landscape description to describe such a gradual change. So in droplet landscapes, we represent on the y-axis the probability of droplet formation, and on the x-axis the multiplicity of binding modes, essentially the likelihood that the binding mode of the protein is going to change if conditions are changing in a cell. So you can see the three different regions of TDP43. The residues that are far from the amyloid core and tend to form droplets, they have very high droplet forming probability, but very low multiplicity of binding modes. So they strongly prefer these disordered interactions. You can see that the distribution is pretty narrow. In contrast, the residues that are part of the amyloid core, they have slightly lower droplet forming propensity, but they are still capable to form droplets. However, the distribution of the binding modes is very wide, so they have a huge increase in terms of the MBM. In between the two, we find residues that are flanking the amyloid core. These residues prefer disordered binding mode, but they have a wide distribution of possible binding mode, so they have the probability to change from disordered to ordered binding, and actually they can serve as aggregation hotspots. If we make this landscape kind of abstract, you find the droplet state here again at high droplet probability and low multiplicity of binding modes. You find the amyloids here with lower probability of droplet formation and high NBM values, and the native state is in between the two. Now, if you make a mutation in a protein that shifts the sequence towards the upper triangle, then the protein is going to aggregate through a liquid state through the condensation pathway. If the mutation shifts the protein towards the lower triangle, in particular to this region, then the protein is going to aggregate through the deposition pathway. So let's try to apply this. We used ALS-associated mutation of various proteins, which were also shown in vitro to induce aggregation. The mutant protein are shown by these red dots and the corresponding Y-type sequences by green. You see that most of the shifts that are occurring in this droplet landscape are kind of horizontal. There is no significant decrease in the droplet probability, but there is a considerable increase in the multiplicity of binding modes, suggesting that these mutations basically expand the repertoire of the binding modes and bias towards sampling ordered bound configurations. If we plug in these principles into a predictor, basically we can distinguish mutations that are ALS associated through inducing aggregation via the condensation pathway versus the mutations that are non-ALS associated. And maybe worth to mention that these include also mutation of very, very similar kinds, let's say G to E, with only a few residues apart. So this may be a powerful technique if more data is accumulated on these ALS mutations. Another possible application of the droplet landscape is to estimate cytotoxicity. So when droplets mature, there are intermediates that are cytotoxic, and actually cytotoxicity is responsible for the neurobiological effects. So in case of TDP-43, using deep mutagenesis, cytotoxicity was estimated for many thousands of mutations. And if we use these parameters from the droplet landscape, we could obtain a fairly good estimate of the experimental toxicity change, both for single mutants and also for the double mutants, which tells us that this droplet landscape description serves probably pretty well to describe what is happening along the condensation pathway. So I try to tell you that protein sequences encode the condensates, both the droplet state and the amyloid state, in a rather universal and context-dependent manner. And this is something that we can predict fairly accurately. Yet the most exciting adventure is still ahead, which is to relate protein sequence to cellular function through this fine-tuned balance of cellular states. So with this, I'd like to conclude and thank in particular Michele Vendruscolo from Cambridge, whom I did most of these projects, also Hao Wu, with whom we started to think about higher order structures. The energy landscape framework was used in collaboration with Stefano Gianni, Diego Ferrero, and Peter Wolynes. We did quite a bit of work on the mathematics of context dependence based on the fuzzy set theory with Attila Horváth from Canberra. We also applied the droplet landscape framework to engineer several cellular processes, among others, muscle cell differentiation. This was done in collaboration with Serena Carra in Modena. We are carrying out very exciting condensed matter physics applied to condensate with Amos Maritan from Padova. And all these tools that you can use for condensate predictions were developed together with Silvia Tossato's group in Padova. Thank you very much for your attention. And now it's my turn also to lead the question and answer session. And I see one question already, but please type your questions into the Q&A. So I just briefly read. Can you define the fuzzy concept of hydrophobic interaction? How do you measure these interactions? Well, there is the fuzzy interaction database in Padova. They released all these different biophysical methods that you can use, including also hydrophobic interaction. So the point is that whenever you bind, you don't bind with well-defined residue A to residue B, but you make the interaction between a set of residues to another set of residues on the other molecules. And I must say that using the energy landscape framework, we started to decipher the origins of specificity in case of such suboptimal interactions, but it's more yet to come. Okay, I read another question. What is the role of chaperones in droplet formation? It's a very, very good question, because it's important that you maintain the structure, because if you start exposing hydrophobic motifs, then you go through the deposition pathway, right? The droplet, the condensation pathway is done somehow differently. It's not necessarily through these hydrophobic patches. And now it's my pleasure to introduce the next speaker, who is Rosana Collepardo-Guevara. She is a professor of computational and molecular biophysics at the departments of chemistry and genetics of the University of Cambridge, UK, and also Vinton Advanced Research Fellow in the Department of Physics. Her group develops smart display models to investigate liquid-liquid phase behavior of proteins, DNA, and chromatin. Rosana received her undergraduate degree in chemistry in Mexico, and then she obtained a master's and a PhD degree in theoretical chemistry from the University of Oxford. Then she did a postdoc with Tamar Schlick at NYU, and then she was a Marie Curie Fellow with Modesto Orozco in Barcelona. She joined Cambridge University as a postdoc with Dan Frankel and David Wales, and where now she is a full professor. So Rosana will tell us how liquid-liquid phase separation regulates chromatin organization. Rosana, stage is yours.
Thank you. Thank you, Monika. Thank you so much for inviting me. I'm really happy to be able to share our research today with everyone. So one of the main research interests in my group is investigating phase separation and also chromatin organization at the nanoscale. And this is a problem in which protein disorder is very important, as I'll try to summarize throughout my slides. And trying to understand genome organization is a question that has gained a lot of attention in the past few years because, in part because genome sequencing has become so efficient that it can now be used as a routine tool, which has enabled now the challenge of trying to interpret our genome, so finding out how it actually functions mechanistically. This is a very hard problem because of many different reasons. One is simply that our genome is huge. If you were to take out the DNA in one of your cells and stretch it out, you would see that it's over 2 meters long. Yet it has to compress enormously to fit into the tiny nucleus of cells, which are almost 1 million times smaller. And this condensation process is extremely complex and important. And just to put this in context, I always like to share this really cool analogy that I heard from Caroline Luger, which is that this extreme condensation and organization is more or less equivalent to taking one human hair. Imagine how thin that is, but imagine that is a 20-kilometer long human hair, and then trying it to condense so much to make it fit inside of a golf ball. And not only that, but also our cells face the challenge of trying to efficiently search for a specific little region of your hair while the hair is still inside the golf ball. So a very hard problem. And not only that, but like folding a piece of paper in different ways to obtain, let's say, a bird, like in my example, or a boat. Different ways of organizing our DNA information are suggested to give rise to different functional outcomes. So in very simplistic terms, we can say whether organizing our genome in one way would give rise to one type of cell or a different type of cell. And the question that we are interested in our group is understanding how different ways of organizing this same DNA information emerge, and how is chromatin organization shape and maintain, and what is the role of phase separation in that. And we are again focused on biophysical and molecular mechanisms. These are actually like kind of big picture questions. They are not exclusive to us. Many other groups are interested in these questions. Because they might be related to enable our understanding of what happens in normal conditions like development or cell identity, but also what happens when things go wrong, like in diseases like cancer or during aging, for instance. So in order to begin to understand these questions, I'll quickly introduce to you the building blocks of genome organization, which are star players in most of our research. So if we were able to zoom in into this condensed chromatin and this condensed genome that is known as chromatin, we would see a few things. One is that the DNA is not found free. It is always complex with proteins that are known as histone proteins that have a global positive charge. That makes sense because the DNA is a negatively charged polymer. So in order to compress it so much and fit it into this tiny nucleus, using positively charged protein helps. So it forms these very amazing 10 nanometer nucleosome particles that I briefly mentioned that have a few very important characteristics. One is that they have 10 disordered histone tails they act like flexible arms. They extend out of the nucleosome surface and allow nucleosomes to contact distant DNA regions or recruit a wide range of proteins, so enabling this condensation and selective organization. However, how this happened, how the selective organization of the genome emerges, is not really well understood at the moment. And one of the problems is that we lack actually close-up views of the molecules that make this condensation process possible. Indeed, this problem of how our genome is organized, chromatin organization, has been resolved at the moment in two limits. One is the limit that I'm showing you here at the top left of very high resolution, near atomistic resolution, but quite small system sizes, so up to a few nucleosomes, or the limit of functionally relevant systems that are larger than the sizes of genes, but at relatively low resolutions. So here on the bottom right. However, the key chemistry of how epigenetic changes and the makeup of the different molecules that form the genome give rise to this organization happens right here at the blind spot that I'm showing you here. And actually, experimental techniques are advancing enormously at the moment. They are reaching the blind spot, which is, I think, very exciting. However, all these methods rely on computational modeling to propose structures that can match the experimental observables, which means that advancing computer models is also really important. And this is the area where my group is trying to contribute. We are developing multi-scale models that are trying to take advantage of the best of different worlds. On one side, as I'm showing you in the slide, very detailed atomistic models of small systems like up to dinucleosomes. And on the other, increasingly approximate descriptions of these systems, but that allow us to get to very large system sizes, including phase-separated domains that contain chromatin, like in the figure here. One of the issues in the field is that for many, many years, experiments of chromatin were only performed in vitro, and they suggested that these polymers of nucleosomes chromatin, when small systems, would fold into very well-ordered structures, a fiber, regular fibers, or more or less than 30 nanometers in width, called the 30 nanometer fiber. Therefore, the hypothesis was that chromatin inside cells perhaps form these ordered fibers as well. And a lot of the models connecting function to structure rely on the interpretation of these fibers, like condensed fibers meant transcriptional silencing, for instance, and loose systems were interpreted as DNA accessibility and therefore transcription activation. However, when more recently experimental techniques advanced to the point to begin to see signatures of these chromatin systems inside cells, they actually struggled to see any signature of disordered fibers. And instead, they began to suggest the model of a more disordered organization, which we are going to talk about in a bit more detail, which is known as the liquid-like model of chromatin, or it has different names. And actually, this is not entirely surprising because in vitro experiments actually needed to be done with perfectly uniform chromatin, in which all the nucleosomes had the same chemical composition or the DNA was the same, et cetera, et cetera. And we have actually shown that this neglect of heterogeneity is an important source of error in the predictions of chromatin structure. When we use a coarse-grained model and we treat everything in the chromatin the same, so a homopolymer, all the nucleosomes with exactly the same chemical composition, all the DNA having the same properties, the same lengths, we predict as well very ordered 30 nanometer fibers. And the moment we begin to introduce heterogeneity, for instance, in this model, something that is very simple to do is simply introduce non-uniform linker DNA lengths, the DNA that connects nucleosomes. We already see a transformation into a polymorphic conformational ensemble. Now, one of the newest ideas that attempts to explain how chromatin might be organized in space without relying on this picture that I mentioned of the 30 nanometer fiber, meaning silencing, is actually the idea of liquid-liquid phase separation. And it's an idea that has exploded in the past few years. And there's a body of work that now suggests that phase separation may actually have many functional implications. For instance, going from the very famous examples of gene silencing by phase separation of HP1 all the way until the other side of the spectrum. We can also hear about the formation of transcriptional factories, which are multi-component phase-separated domains promoted by transcriptional coactivators, some proteins found inside the nucleus. And not only that, but from the more physical-chemical point of view, which we find very interesting, the Rosen lab found that the chromatin polymers themselves can also undergo intrinsic liquid-liquid phase separation without the aid of any additional proteins. So motivated by this, we wanted to investigate the driving forces for this chromatin phase separation. And for that, we developed a multi-scale model, kind of what I showed you at the beginning, linking atomistic properties of nucleosomes to a coarse-grained model where we consider all the amino acids explicitly, all the different DNA base pairs explicitly as well. And then finally, a minimal model where is derived from the higher resolution models that we have developed, but now we can represent a nucleosome with just a few particles and achieve these simulations of chromatin phase separation. So one of the interests that we had was trying to model nucleosomes with increasing accuracy or realism. And we wanted to capture a key observation, which is that nucleosomes are actually not rigid building blocks of the genome, but they are highly dynamical and heterogeneous particles. And thermal fluctuations cause some of these DNA histone core interactions to spontaneously break and reform in a phenomenon known as nucleosome breathing that I will be mentioning a lot in the next few slides. And to capture this, we developed this relatively high resolution model that I mentioned, where all the amino acids are represented explicitly and the DNA base pairs as well. But kind of the important ingredient is that the DNA is not bound permanently or artificially to the histone core, but rather this binding emerges naturally from electrostatics and hydrophobic interactions. And also the mechanical properties of the DNA are very important. Now, reassuringly, when we use our model to measure the response of nucleosomes to force as they unwrap, we get excellent agreement with experiments where they have performed the same. So unzipping experiments at single base pair resolution, which suggests that our parameterization is reasonable, which is good. Okay, so now we can use this type of models to investigate phase separation, which is what we were aiming for. That's why we developed these models. And importantly, considering these breathing motions, which is what we were after. And with this type of simulations, what we can do is compute phase diagrams, as Gerhard just mentioned, which allow us to quantify the range of parameters that we are choosing that favor phase separation of chromatin, which is basically in this cartoon, this shaded area here. And in particular, we are investigating for this system, the space of salt concentration because chromatin density, because it's relevant for the experiment. And we investigate different parameters, but first for validation, we focus on this changing the length of the DNA between nucleosomes because that was done by our experimental collaborators. So when we first investigate these lengths of in the series 10n+5, so that means 15, 25, and 35 base pairs, and we measure phase diagrams for chromatin with these different parameters, we see large coexistent regions, which are in perfect agreement with the results of our collaborators observing droplets forming easily. Now, when we use DNA linkers length equal to 10 and something, so 20 and 30, we see a small coexistent regions also in agreement with what our collaborators see of not droplets forming easily. So this is just the validation stage. It shows that our model is capturing the driving forces for chromatin phase separation intrinsically in a decent way. So now we can go and use our model to try to gain some insight on these nucleosome breeding motions. So what we did was simulate phase separation of chromatin in two cases, one allowing these natural breeding motions of chromatin, and then as a control case, constraining these motions just simply by artificially gluing the DNA to the histone core, so inhibiting those motions. And we discovered that these breeding motions are actually critical in promoting the phase separation of chromatin. So we observed that this curve in red here gives rise to a larger coexistent region, which is for breeding nucleosomes. Now we wanted to investigate why actually these breeding motions enhance chromatin phase separation. And what we can do is perform simulations using our models, and we measure in the resulting equilibrium configurational ensembles the frequencies of interactions between first nearest neighbors, second nearest neighbors, which I'm showing you here. This is the frequency, this is the how far the neighbor is within a single chromatin fiber, and we break it down by orientations. So whether the nucleosomes are well aligned face to face, this is the orientation called like that, face to side or side to side. So what you are seeing here is basically that when we allow these breeding motions, you can get a wide range of different interactions and orientations. The interactions are heterogeneous. And this is consistent with this idea of chromatin having this liquid-like behavior. In contrast, when we do our control, we artificially glue the DNA to the histone core, we inhibit the breeding motions. We now see that mostly all the nucleosomes interact with the second nearest neighbors face to face, as you are also seeing in the little video there, which is a typical organization of this 30 nanometer zigzag fiber. Now, so why does this matter for phase separation? So then what we did next was do a bit more simulations to try to understand how these liquid-like versus rigid fibers impact phase separation of chromatin. So what we can do with simulations is, for instance, do pooling experiments, take single chromatin fibers and just apply force and estimate the energetic cost of breaking the interactions in the different fibers. So we see that the energetic cost of pulling or unfolding some of these rigid are not allowed to breed is high compared to the ones where nucleosome breeding is actually allowed. And more interestingly, when we now compute the potential of mean force interaction of two of these fibers by umbrella sampling simulations coupled with replica exchange, we see that in the case where chromatin is liquid-like, the interactions between two of these fibers are much stronger than in the case where chromatin is rigid and static. And that makes sense when two liquid-like chromatin fibers meet, they kind of melt and they form a lot of interfiber interactions with other systems. So again, favoring phase separation. So one of the other things that we can get is trying to understand what is the role of protein disorder, which I promise I'll mention quickly about in promoting this more liquid-like organization of chromatin. So with our models, we can zoom in into the interactions that are actually determining the different types of structures of chromatin. So I will not go into detail about what you are seeing here just for the sake of time, but basically we can interrogate with which specific amino acid interactions or amino acid DNA interactions are more frequent in the different systems. And we see that in the liquid-like chromatin, the majority of the nucleosomes interactions are mediated by the disorder histone tails and the DNA. So all the histone tails are playing a very crucial role in stabilizing this type of structure, whereas in the zigzag fiber, the most dominant interactions are among globular domains and globular domains and globular domains and the H4 histone tail. Okay, so another thing that I want to briefly mention is that this protein disorder, by mediating these interactions, also increases the valency of nucleosomes. So when we compute for each of these fibers, how many other nucleosomes they are interacting with per unit of volume, we see that for this breathing nucleosome system, we have a higher valency, a higher number of contacts. This is just salt concentration. So another thing that we are observing here is that the valency of nucleosomes is very context specific, which is something that you would expect if you have lower salt, they form less contacts, more electrostatic repulsion among the DNA. If you have slightly higher salt, they form more contacts, right? So the interesting part is that this connection between valency and stability of condensates is something that is not exclusive to chromatin and nucleosomes, but it's something that has been well documented by many people, Shortino, Rosen, Rohit Papu, Tanya Miktak, a lot of people in actually stabilizing condensates. And it's not surprising that it's also a feature that emerges when we use patchy colloids, so simply patchy particles to simplify the description of proteins and try to get this insight on what's happening. What we observe with these very simplified models is that a higher valency gives rise to a more densely connected liquid network, and the more connections, the higher the enthalpic gain for condensate formation, so it makes sense. And now, like I mentioned before, this variability of a nucleosome valency with the salt concentration with the environmental condition, again, is also something that emerges in protein condensate, not only in chromatin, and it's something that was highlighted by modeling experimental collaboration that we did recently. We observed there's a strong modulation of the phase behavior of proteins in general by salt concentration. So our experimental colleagues discovered that certain proteins can undergo salt-mediated reentrant phase separation, meaning that they can form condensates both at low salt concentrations, like shown you here in the slide, then they dissolve, and then they phase separate again at very high salt concentrations. And what we did was combine atomistic simulations with coarse-grained simulations to try to identify the different driving forces for condensate formation at low salt and high salt. And we observed that the different salt regimes, in the different salt regimes, different types of molecular interactions dominate. And also, again, the valency of the molecules changes depending on the context. So kind of the point that I want to highlight here is that the ability of a protein or a molecule to phase separate is not only encoded by the sequence, but is highly modulated by the conditions and the environment where the protein or biomolecule it is, including the composition of the system. So if we immerse the same protein, for instance, in this example, HMGA1, in a system where now it has a different binding partner, the valency, the density of the phases, its ability to promote a phase separation also change. Okay, to close, I just want to say that we have developed now recently a coarse-grained model for biomolecular phase separation for proteins that achieves really good agreement with experiments and is widely available. So if you are interested in modeling phase separation, you can get in touch or like download it from our GitHub. And we did some benchmarks against quantitative experimental phase diagrams for the protein HNR-MPA1, and we get really good agreement with the actually critical parameters, which I think allows us to begin to understand how mutations in proteins actually shift this phase boundary. Okay, so to conclude, I just want to mention that we think that this ability of nucleosomes to modulate the phase behavior of intrinsic chromatin condensates without proteins may actually also extend beyond the formation of condensates that only contain chromatin, because there's many proteins that phase separate that can also modulate the formations of nucleosomes. For instance, the protein HP1, one of the most famous, that allows the formation of heterochromatin condensates, has been shown to deform nucleosomes when it binds to them and promote phase separation at the same time. Okay, so to close, I just want to thank all my students and postdocs for the work. Everything that I show you was done by them in particular, Gerald Joseph, who is now a PI at Princeton, so she's just started. I'm really proud of her. Jorge Rene, who is here, and Steve, who developed our corresponding models. I don't have him in my picture, but his picture was with us before. So, okay, that's it. Thank you, everyone. Thank you for listening.
Thank you so much, Rosana. And I think we have time for one question. And let me read this from Mihai Mezei from New York. So have you seen in the simulations a histone core sliding along the DNA chain by staying encircled by the DNA?
Yes, actually, that's something that I always forget to mention. But yes, in our model, we because we are only allowing the formation of nucleosomes via normal electrostatics and hydrophobic interactions, we see both breathing and sliding. And that's something that we actually have never published, but sometime we will, like, we have all the analysis of how much the nucleosomes move. And it's also something related mostly, at least in our simulations, to the sequence of the DNA. So how mechanically rigid or torsionally rigid different DNA sequences are. So, but I guess it will also be contributing to promoting phase separation if it does the same as breathing, like the forming nucleosomes, enhancing the valency and the regularity of the structure of chromatin, which seems to be critical for the formation of these condensates.
Okay, so all those who would like to ask more questions from Rosana, it seems there are a few others. Please stay until the end of the session and we will try to answer these. So it's my pleasure to introduce the next speaker, who is Peter Boliness. He's a Bullard Welch professor at Rice University in Houston and the co-director of the NSF Center for Theoretical Biological Physics. He applies a wide range of techniques from quantum chemistry to coarse-grained methods to understand how protein structures are formed. He's a pioneer in the protein folding field, very well known from the energy landscape framework. Actually, his CV is very challenging for this kind of webinar introduction. So I try to cope with this. So Peter obtained his PhD at Harvard. And at the same year, he became also an assistant professor in Harvard. Later in 1980, he moved to Illinois, Urbana-Champaign, where he became an Eisner Professor of Chemistry and Professor of Chemistry, Physics, and Biophysics at the Center for Advanced Study. In 2000, he moved to University of California, San Diego, where he was a Francis Crick Chair in Physical Sciences, and he took his current position in 2011. He also had external appointments like a lecturer in Oxford as well as in Cambridge, and he was also a scholar at the NIH. Peter has a very long list of awards by many societies: American Chemical Society, award in pure chemistry, Peter Debye Award for physical chemistry, award in theoretical chemistry, biological physics prize by the American Physics Society, and he was elected in 91 to be the member of National Academy of Sciences and the American Academy of Arts and Sciences. In addition to that, he is also a member of the American Philosophical Society. He is a member of German Academy of Sciences, a foreign member of the Royal Society and Indian National Science Academy. I hope I did not forget anything crucial, but anyway, it's a pleasure that Peter accepted our invitation, and he will address a very challenging topic in neurobiology, ranging from prion simulations, bacterial channeling. Looking forward, Peter.
Well, thank you. It's a very nice, I think it's, sorry that the CV is long. Just so that my Indian colleagues are not upset, I should say that I'm also a member of the Indian National Science Academy, which is also an important organization. So thank those guys. Okay, I think let's see, I should be able to screen share, is that right? If I press the button, share screen, and I do.
Okay, we'll be able to see it. Yes, we see it. Just
Yeah,
put to
see it?
projection.
Okay. And then let's see, what next do I need to do? I need to make sure that I move it over and do play from start.
Okay, perfect.
You guys. Okay, well, thanks very much, Monika. It's been a pleasure actually also interacting with you and hearing about this emerging story of fuzziness and protein condensates. I think it really is an important enriching of how we view the biochemical world. I'm going to tell you about just a couple of problems we've been looking at that have been inspired by thinking about the problem of, what's the origin of memory and how do neurons work? I got into this problem by two paths. One was that I had a student, Ming-Chen Chen, who is a great research student of mine, who's now running his own company back in China. And he's a great guy. And I started him out on a starter project, which was to look at prions that were involved in the formation of memory. And I will be telling you a little bit about the part of that story later. Now, it turned out that these prions that are involved in forming memory do so by interacting with the cytoskeleton, which was a problem I'd already been working on with several postdocs. And then in addition, Margaret Chung, who's a member of our center who recently moved to Pacific Northwest Lab, was collaborating with neurobiologists on sort of the initiation of memory events involving CAM kinase. So we sort of joined forces partly with Margaret on that part of the problem. And what we found is it's an incredibly rich problem that brings us into sort of new physical questions. And I was going to tell you about a couple of them. I think they're at least relevant to the condensate question, although the first part is not specifically a condensate as we're now thinking of it, condensate of fuzzy proteins, but condensates of more organized proteins involved in the cytoskeleton. And then I'll go on to why should it be that forming a structured object should have any influence on sort of molecular biology problems in the sense of regulating translation. So hopefully this advances, yes. I got to be a little careful why, okay. I'm screen sharing, okay. So just to fill you in on the sort of basics as we know them, although I've got to say this is an area that is still emerging. It seems to be true that basically the way in which memories are stored is something like a scheme based on associative memory. And in fact, there's a lot of physics of associative memory that was used, for example, in setting up the energy landscape theory of proteins and protein structure prediction. But the basic idea is if you keep using the same thing over and over again, it becomes easier to use. And how that's implemented in neurons is, so for example, in protein folding, how that's implemented is if you keep seeing interactions between individual residues like hydrophobic residues or something like that in lots of structures, you should make that interaction a strong interaction in your model. But brains are much more complicated than protein problems. And instead what we have in brains is lots of neurons, and the neurons are connected to each other by things called dendritic spines. And if an electrical signal is put into the dendritic spine, then it actually causes the spine to grow. And it does this by reorganizing the cytoskeleton in response to these electrical signals. And this in part comes through the introduction of calcium and gets into the Margaret Chung part of the problem, how calcium binds to things like CAM kinase and how that reorganizes the structure of the cytoskeleton. So part of this is how can a cytoskeleton even move and move from one place to another? And there probably are many mechanisms doing this, but it's one that we started to look at in a more general way. And in fact, of course, for many years, this has been a problem that has attracted biological physicists. And it's much easier to study in vitro than it is in vivo, but of course you don't know whether you have all the right ingredients, and you certainly don't have the regulatory apparatus. But this is an example of a study now, I guess, getting to be 14 years ago by David Weitz, and similar work of Fred McIntosh, my colleague here, where you make a cytoskeletal assembly out of actin, out of motor proteins, and out of, in this case, a protein called actinin, which acts to bind together actin chains. And one of the interesting things here is that at low concentrations, nothing much happens. You form kind of a very small microscopic size droplets. Above a certain size, you start to form something that looks relatively solid, and it's solid and able to contract on the basis of motor motion. And then actually when you put in too many crosslinks, it starts to it becomes hard and unable to contract. And this is a kind of phase diagram that one gets that depends on the concentration of the sort of generic cross-linking proteins on the bottom axis and on motor proteins on the top axis. And you need to have both motors, and you need to have cross-linking by other proteins as well, to have a material that can actually be, that can actually move, and therefore, for example, move into that dendritic spine. So one of the ways in which this can be modeled, and has led to a very large number of good ideas, is using detailed codes of the assembly of different parts of the actin and cross-linking molecules. The leader in all those sort of developments is Gerhard at the University of Maryland. He also works with us on problems in protein structure prediction. But in this project, he's been the big guy and done all of this mostly at Maryland. Well, actually all at Maryland. He has a code called Median. And in this model, one actually sort of assembles actin itself out of individual parts. They stochastically bind. They introduce mechanical forces on each other. You also can have cross-linking proteins, and it's a very general way of looking at binding problems. Now, we're going to take that to start with as basically the ground truth. And one of the things we asked was there a simpler way to understand this? And is it possible to understand the connectivities and how they change with time and how they affect the mechanical properties? And this is a work of my student, Carlos Bueno and James Lyman, also involving Margaret, where we compare the results of simulations based on median of actin networks with cross-linkers and motorized things to a model that's just chemical kinetics. And what are the species in the chemical kinetics? Of course, there's tons of different length chains and things like that. Actin both assembles at one end and disassembles at the other end, so-called plus and minus ends of the actin. So there's tons of different species, but you can use a kinetic model that just asks, what's the kinetics of forming connections? And what's interesting is a model that is sort of a very simple chemical rate law model for the probability of forming connections, gives essentially the same results as a rather sophisticated simulation based on median, and tells us at any given time, after you start mixing the things together, how many crosslinks of different types that you have. And it begins to fail, excuse me, at some point. And the interesting question is, why does it fail? And it actually doesn't fail by a lot, even when you get to this point where the kinetic model, simple kinetic model, breaks down. So what we did was ask the question, how connected are the objects that one forms by making all these crosslinks? The chemical kinetic model tells you what's the probability that a certain crosslink actually is formed. As I said, there's crosslinks that form even the actin itself. They are things that take an actin filament and put another G-actin onto it, and another G-actin, another G-actin at the plus end, or I forget whether it actually binds at the plus or the minus end. It dissociates at the other end in these models. That can, of course, be out of equilibrium, and that's how you get things like net motion and treadmilling. So the question is, when you know all the connectivities, where everything's connected, what sort of object do you get? And is it something that fills the whole system? Is it something that's fluid, or is it rigid? And we use an idea that goes back to Flory and Walter Stockmar, basically actually in the 40s, which just asks on a sort of network that doesn't live in 3 dimensions, but lives as an infinite tree, the so-called beta lattice. If I tell you what the connectivities are, count the number of different species that I have, and ask the question, when do you have a species, which corresponds to percolating through the entire system, a globally connected macroscopic material. In a sense, that's the same thing as a phase separation. It's not quite exactly the same. You need a little bit of extra oomph from some residual forces to get phase diagrams like the ones we just saw from Rosana. But basically, it tells you the beginning of that phase separation process. And one of the things then is one gets a phase diagram that depends on how likely is it that the chain forms connections in the actin. So that determines the lengths of the actin fibers. And then also how many other kind of cross links do you have, which depends on these other ingredients. Now, one of the things in that phase diagram is that, as well as how often do you have motorized connections. And you can see that either kind of crosslink actually has a tendency to form the thing into a connected cluster. And this is a picture of the phase diagram that results from that. Now, what one finds is if all those connections were absolutely rigid, then the object that you would form that's connected in that way is also rigid. If some of those connections are floppy or disordered, then you do remove degrees of freedom, but not quite as many degrees of freedom when things bind. And so you have to add many more crosslinks before the material begins to become rigid. So these are the sort of 2 different transitions in the behavior of the system. One is to a very fluidy but still connected object that phase separates, and another one is a rigid object that behaves like a glass, has aging properties like Gerhard was talking about, and flows in a very, very, very different way. So what one finds is that you need to have both the percolation of the, you get effects that come from percolation with the motors, and also percolation due to the linkers. If you have a sort of intermediate level of those, then the motors are able to pull the system. It has to be globally connected before it can pull. But if it's over-connected, the system becomes rigid. It undergoes that glass transition that we were talking about. And that will happen if you add in too many linkers. So this is sort of the same kind of thing that we saw in those experiments of David Weitz and Fred McIntosh. So what happens when you're near that transition? How do things move? That's how we think is sort of at least the beginning of motion into those dendritic spines. Turns out that once you seize up overall, but you don't completely seize up. Instead, the system still very slowly changes, but it slowly changes through little earthquakes, or I guess some people call them cytoquakes. And this is a picture of what happens in a simulation using the Median code from Gerhard when you're in that region where you're very connected, but this is still a dynamic system. The actin chains are still doing their thing. The motors are motorizing. Systems overall rigid, but you sometimes have these motions that amount to little parts of the system, little compared to the entire simulation box, but that involve huge regions reconfiguring. These occur very, very rarely, however. So so this is a little bit like the way things happen in the motion of tectonic plates on the Earth. That every now and again you get an earthquake and the plates slide. So this is, I think, first of all, one of the mechanisms that I think is going to be playing a role in general in how condensate systems move when they're very dense, and of course is involved in this rearrangement of things in dendritic spines. Now, as I said, there was this other path into the problem that started with Ming Chen, which was involved with the question of, well, how do you get truly long-term memory? Things we were talking about there were being simulated. They involve processes on the order of minutes to hours. But of course, when you form a memory, that memory seems to last for decades in some case, if you're lucky, or I guess unlucky if it's traumatic. But we start to worry about losing our memory at my age. But one of the problems that Francis Crick, I think, first pointed out in the 80s is most proteins don't last that long. So this is one reason why you have to have structures that are very long-term. And one of the candidates for forming these structures is candidates that involve prions. Eric Kandel and Susan Lindquist identified a protein that was involved in forming prions. It was called CPEB. CPEB ordinarily, when you make it in vivo, forms sort of droplets, but to form a memory, it has to form a fiber. And the first part of the story that Ming Chen worked on with Weiwa Zheng and me was to sort of understand how could that thing change from a droplet into a prion fiber. And Ming Chen realized that if you pull on an alpha helix, which is the nature of the prion protein by itself, it has much more of a tendency to form a beta sheet. And in fact, we think that the prion forms by being pulled on by the cytoskeleton in one of those earthquake events, probably. Now, what's interesting here is that the protein, when it forms, when the prion, when it forms, changes its ability to affect the translation of other proteins. And in fact, one of the proteins that it causes translation to enhance is the messenger RNA for actin. So you can see that there's kind of a feedback cycle here. You would have something where you make CPEB fibers. CPEB fibers recruit actin out of actin mRNA. There's more actin, and so now you can keep building the cytoskeletal assembly. So a question that arises there is, why should CPEB in one structural form, sort of a condensate form, not particularly encourage the translation of mRNA? It always binds RNA, it has the potential to bind RNA, but it does so much more readily in this once it's formed a prion. And my student, Xinyu Gu, working also with one of my ex-students, Nick Schaefer, had a really great idea that this has to do with the structure of the prion and how the structural details influence the ability of the RNA that binds to the CPEB to be translated by ribosomes. So the picture is this: that in the prion, you have a bunch of RNA binding domains hanging off of the central prion fiber, and we know where how the RNA binds to it, and we also know then where the ribosomes begin to translate it. It turns out that there's a lot of proteins that are involved in condensates that are RNA binding proteins. They also often have the signals to form prions. There's another example of this, a protein called RIM4, which also binds. And what's interesting is RIM4, when it forms the amyloid, represses translation, but CPEB, when it is in the prion form, enhances the translation of the RNA that binds. And it turns out the difference between these two things is whether the starting end, the 3' end of the RNA where it binds, is far from the ribosome, or whether it's in the central part of the ribosome. And what you can see is that if you have something like the situation where we think about enhancement, a ribosome can bind on the outside, can be translated down the RNA fiber, and then is released. And it's likely when it's released that it will bind to another one of the fibers and sort of cycle around. So the prion is a factory for translation. The opposite thing happens if the 5' end is buried close to the prion fiber. Because then, in the opposite polarity, because in that case, the ribosome has to make its way all the way inside, and that's hard. It has to go through this sort of fuzzy covering, and then, if anything, it's led to the outside where it has more of a chance to escape. So, so this is why there's a phenomenon which we call channeling, and we call it vectorial because it depends on the directionality. I guess this is a slide that just sort of goes over that that idea. And Xinyu analyzed this with a kinetic model. The kinetic model basically talks about the proteins binding at the start codons by a straight diffusion process. Of course, they become translated at a net rate down to the location of the prion end in the case of CPEB. On the other hand, once that means they're deposited at that point, there's a source of the ribosomes, and then they diffuse away, and when they diffuse away, they then get a chance to be captured by a different CPEB molecule and with an RNA attached to it. And this is the kinetics of that diffusion process, with a source at the central region and a sink on the outside of the factory. And one can work through what happens in the steady state. You get an interesting kind of steady state diffusion law, which is non-local. And this can be solved by some mathematical tricks. And this is the result. That in the case of CPEB, you find that you get modest enhancements that depend on how much harder is it to translate through the, for the ribosome to translate through the fuzz around the fiber. On the other hand, in the case of RIM4, you get really, really strong repression in this case. So I think one of the points this makes is that the actual structures of the prions matter in terms of whether you get large effects or small effects. And in these models that I'm showing here, there's no particular, say, stronger binding of ribosomes to the fuzz in either case. If you have some additional strong binding to the fuzz, even moderately strong binding to the fuzz, these effects become much larger, considerably larger. So I think this tells us that, of course, the structure of prions and the fuzzy fibers around the prions are probably very important in their function. And likewise, I think in the condensates, one can have questions of, are there local polarized structures that change how RNA is transcribed? So I think this puts a lot of reason to start to study those systems much more carefully and structurally, and not assume that a condensate is just a liquid without any particular structure. Of course, other people have been saying that from other experiments and reasons show that condensates, even though they look superficially like a liquid, they often have a surface layer that's different from what's inside. And in fact, the amount of enhancement in the case of CPEB is reasonably well in agreement with experiment when one has just a little bit of this attraction. And again, the RIM4 diminution effect is quite considerable. And here, of course, the CPEB, when it's in the condensate form, turns out to be have just the effect of the slower diffusion, and that's why it's actually represses in that form. Okay, well, I just want to end there. There's many, many more questions that are implied by thinking about neural memory, about how proteins actually get together, how they move and do things. That's why we have we have a group here at the CWP devoted to that we call it the memory group. It's been funded, partly it was funded by this special grant from the NSF, dendritic spine mechanobiology. Herb Levine, who is from our center, recently moved to Northeastern, has been part of these efforts. Margaret Chung, our experimentalist Neil Waxham worked mostly on the CAM kinase questions, which I didn't talk about. Carlos Bueno and Ming-Chen Chen are key, first four people are the key players on this part of the problem. The Shinyu's looking for postdocs, and James has got a job, and Ming-Chen's already making millions in his company. So with that, I'll go ahead and take questions.
So thank you so much, Peter. I really enjoyed showing the different sides and also kind of this double-edged sword thing with the multivalency that balances between condensates and prions. So the first question, well, it's to all speakers, but maybe you can start answering it. Dynamics of watering condensates versus aggregates or prions. I think this can be related to the structure, specific structure in condensates that you were mentioning. It was asked by Dennis Renard.
Okay, I don't know if that's to me because I didn't talk too much about that microscopic level. We actually do things with this code we call AWESOME, which is the Associative Memory Water-Mediated Structure and Energy Model. It looks at the short range and sequence things and uses neural networks to get those. Then the long range interactions involve water mediated effects. And I was actually pleased to see in Rosana's talk that she highlighted many of the interactions that are very important in the AWESOME model. So we're using it for a lot of things like that. We've not actually done anything on using it for the condensates. That's a problem for the next round of students. So I can't tell you what's different in the condensates. In the prions, Shinyu has looked at this quite a bit in the particular case of CPEB. And one of the things is in all the, as well as in the actin part of the problem, there's often a problem where a disordered fragment is actually the regulatory domain. And I guess we call, and in fact, it binds often to the actin through water-mediated interactions. A lot of those interfaces are water-mediated. And it's interesting because we've done some other work where we looked at, say, for example, Rosetta, and I was actually shocked to find that Rosetta doesn't have interactions like that in it at all, at least the version that's publicly available. So, so I think that associations through the water are very important.
Thank you so much, Peter. And now I think the floor is open to all of us. And there were a number of questions that we couldn't answer. I particularly failed to answer lots of questions because I was listening to your talks. So the way how I thought to proceed is just to read the questions one after another, and well, all of us can answer. Okay. So I start maybe with the first ones. Okay. Will the inhibition of droplet formation become a new therapeutic mechanism? Julian Lee. Well, there are a number of papers suggesting this, right? But I think until we are not fully aware of the mechanisms, it will be kind of an experimental trial and error. But please do comment on this. Does any of the speakers?
Well, I was going to say that normally, the pharmacology type people, I'm not deeply involved with them, but where drugs have been very successful actually has been in inhibiting enzymes and things of that sort. And they're part of the reason that you can find a drug that binds one place and not another is it has a very specific structure. So there's always been a sense that it's harder to do this, to regulate condensates by trying to bind somewhere special in the condensate, even though that doesn't prevent us from sending to the NIH thing saying we'll look at such questions. I think there's more hope in this thing that Gerhard raised, which is that the phosphorylation does in fact play a key role in forming the condensate in the first place in many cases. And there one can easily think about enzymes, drugs that inhibit specific kinases and things like that. That's a very big business. So I think that indirect way probably is quite promising.
Also,
Yeah, I think, sorry. Yeah, I think another idea that probably has gathered more acceptance rather than actually modulating formation and dissolution of these droplets is exploiting their properties to selectively partition small molecules into condensates. So that might offer as well therapeutic options. But yeah, it's like very far from where the field is, I think.
If
yeah,
I can
also,
maybe,
go ahead.
maybe just add, I think an important aspect is it's very often maybe not preventing the formation or the dissolution of condensates, but very often it is maintaining the condensates in a functional state, which could, for instance, be liquid dynamic with the proper composition. So, and of course, that is almost certainly a problem that requires the regulation of a number of factors that contribute to these properties. And I think, as Peter already said, chemical modifications are clearly one important factor.
I mean, also in forming the amyloids, there are a few weak spots, like, for example, the actual tip of the fiber is something that's a small amount of material. So you could, in principle, bind something there to affect the aggregation. That's kind of something that a colleague, Peter Vekelov here, has been finding. I know Bill Eaton is doing big surveys of amyloids. One of the problems is that for many of the standard diseases like Alzheimer's, there's certainly a long slew of people who believe that the amyloids are good and are not bad. So there's that big physiological biomedical question that's sort of looming out there. Too much amyloid is clearly bad. There we showed, and well, first of all, it's been known for a long time, there's a variety of mutants that give rise to extremely bad early onset disease. Those clearly encourage aggregation when you do simulations of them. And the same is also true of Huntington's. So there clearly are things where aggregates, making them aggregate better makes people worse. So so that's, so I don't think they're always protective anyway.
Okay, so let's answer some of the amyloid-related questions. The first is from Sonia Longhi. How can you reconcile the fact that some amyloids resulting from maturation of droplets, the region that become ordered in the core of fibrils, is rather small, implying that the extent of disorder remains high? Okay, so let me start answering this question. So what you do here is shifting the populations gradually, and this is it's sufficient if you do a minor change. And you start this population shift because that will eventually form a seed. So it does not contradict the fact that amyloids can form through this condensation pathway, only a small part shifting completely to the ordered state, while the rest is kind of adjusting and then providing appropriate context for this process to occur.
I have something to add. Yeah, we have a paper where we use like coarse grain models to try to investigate this together with Thomas Knoth. And what we observe is that related to what Monika was saying, you don't actually need like all the proteins to form fibrils for the condensate to become arrested. What's happening, like if you actually zoom in into the network of connections inside a mature condensate, is that you have the network of connections established by the parts of the protein that have undergone a disorder to order transition, which is not fully percolated. So they need percolating throughout the condensate. So basically, all the points, all the proteins have anchors that are established in long-lived bonds, and therefore the proteins cannot longer freely diffuse within the condensate. But it doesn't have to be all the components of the condensate. If the systems are multi-component or all the protein completely having this fibril-like state, it just has to be a sufficiently connected network where the bonds just cannot longer break within the time scales that you are looking at things.
No,
Sorry?
I was going to say something that one of the surprises I had about 10 years ago was I always thought that the amyloids were sort of originally sort of glassy type things or something like that, or because everyone called them misfolded. And so I never believed any of this stuff that David Eisenberg was saying or Andruskalo was saying about there's amyloidogenic fragments. And then Nick Schaefer did simulations where we found exactly that the thing, we just were doing dimers coming together. We found the amyloidogenic fabric, specifically bound, and we developed things based on that. So I think that that it actually is a key thing that there's only a small part of the protein that's involved in nucleating the amyloid, and probably like in the CPEB case, you're left with this fuzzy exterior, but it might be the fuzzy exterior that's doing the business. Rather than, it's important that everything be brought together by strong interactions, but the fuzzy exterior may be doing the physiological function. So I think that that's at least part of the story. And if it wasn't true that it was a small part, then everything Monika said about I look for little signals here or there, that would have been not have correlated with anything, but it does correlate. So I think we've got to accept that as the way not completely ordered proteins work.
Okay, a tight related question to this: are these droplet proteins essential precursors of amyloid fibers? Well, we know that all proteins form amyloid fibers, but particularly think that droplet proteins kind of the first defense line of protein homeostasis to prohibit this aggregation to occur. Other answers, comments?
I think it can be both ways, and depending on the composition of the condensate, you can have droplets that prevent the formation of amyloid fibrils, and there are others where, like, just having higher concentrations of proteins and the high density fluctuations within the droplets that might promote amyloids. But I think Thomas has shown that, and others, that the fibrils can see that the surface of the condensate, because surfaces are not kind of smooth. They have these, I forgot how they are called, but kind of they are rough if you zoom in into the surface, capillary wave fluctuations, and that also promotes formation of these fibrils. But yeah, as I said, I think it depends on what else is in the condensate.
Okay, are small proteins more likely to form condensates than large proteins? What do you think? Well, I can answer it only from the point of view of binding heterogeneity, which is more likely to occur in small proteins. So in that sense, we can expect. On the other hand, simulation people had shown that long linkers, of course, in particular through modulating different kinds of binding orientation, also promote lipid-lipid phase separation. So I think there could be different molecular factors contributing to this, but definitely this kind of heterogeneous binding is probably one way, and the other way is the degradation, protecting them from degradation, because in the condensates half-life may be longer. Other opinions on this?
Yeah, I think that generally I would expect it to be the other way around. So the bigger the protein, the more binding sites, the higher, the more possible connection points, so the higher the valency. But I think what you said is correct. It would depend on the structure, if it's disorder or not, or like how the binding sites are, whether they are accessible, where there's also like higher affinity for the solvent or for other biomolecules. I think it depends on a lot of factors. But if it's exactly the same sequence, it has been shown that having like multiple repeating domains of the same protein actually promotes phase separation just because you have a higher valency.
Yeah, I would say that it's hard to make an argument because obviously proteins are selected objects, evolved objects to be minimally frustrated. And so a very, very big protein that's naturally very big is filled with minimally frustrated stuff. And so it can be very, very stable and not be able to ever spend any time in condensate form. So that it has more of a chance to have an ordered structure than a little thing. But you can obviously make super long stretches of long intrinsically disordered glycines or something like that. But you know, even in that, for example, in Huntington's disease, right? Huntington's disease, the thing that finally aggregates is a homopolymer, poly-Q. And it's only when it's very long that it finally aggregates and forms fibers. So you can make arguments both ways because sequence can evolve in so many different ways.
Yeah, so I think that absolutely from a polymeric perspective, you would expect long stretches of heteropolymers to have a higher tendency, kind of if all things were more or less random. But in an evolved system, other factors obviously are at play. But it looks like if one looks at biological functional condensate structures, they seem to be they don't seem to be particularly short stretches.
Okay, I got like 3 questions. It seems I'm really lagging behind the questions regarding my methodologies on the droplet landscape, how the intrinsic dimensionality of a droplet landscape differs from the conformational landscape, and also whether multiplicity of binding interactions defined sequentially. And also I got somewhere a 3rd one. Let me group these 3 and answer together how we basically validate the methodologies. So let me just answer briefly, and maybe that would also connect to previous questions, that the dimensionality of the droplet landscape, and formally it's 2 dimensions, right? But there is a hidden 3rd dimension, which is captured by the binding mode multiplicity, which is the effect of the environment. So that's kind of the 3rd dimension. And this is defined consecutively for all the residues. And the way how you derive it, so basically the method validation is done on thousands of protein complexes that are in the PDB, both in bound form and free form, or in bound form, the same stretch in different kinds of binding modes. You find surprisingly large amount of different binding modes for the same stretch. So it's not that difficult to parameterize, in particular that we use 2 or 3 terms only for the parameterization. So it's not a, let's say, tuned or fudged neural network algorithm, but it's a very simple logistic regression that we do intentionally so that we don't depend so much on the data sets. But again, I think the most important point that this is kind of a hidden 3rd dimension that is expressing the effect of the context that is becoming, I mean, in all the talks, I think it was very clear that this context matters and small effects in electrostatics, hydration, linkers, the fuzzy coat and so on. So in all the different aspects, these things really matter. Okay, then they ask us about estimation for E. coli, the kind of the fraction of proteins that form condensates in E. coli. So what would you say about this? Then because we had actually done some calculations, and it's not much different from human proteins. Of course, E. coli contains more structured proteins, but again, the binding interactions and the binding modes can be disordered. And indeed, E. coli contains a huge amount of promiscuous enzymes that probably are somewhat related to this kind of interaction heterogeneity. This is what I would say. Okay, let's move forward. Just a second. Okay, Could be Alzheimer's disease a problem of protein condensates? A problem of protein condensates, a protein like myosin that usually should not be aggregated. So what do you think about this Alzheimer's disease thing?
Well, one of the problems with Alzheimer's disease in terms of funding it, researching it, is there's two completely different camps. One says it's A beta, one says that it's tau protein. They hate each other viciously. And even if you write in your proposal that you'll study both of them, that one of them will kill it. So I think there's still just tons of problems of understanding what's the physiological cause of losing memory. That's part of the reason why we started this memory group, because I kind of feel like if you don't understand how memory itself is formed, then how can you figure out how to fix it? And I do think that's why there's been this focus on cytotoxicity. It might not be cytotoxicity. It might be that the formation of one kind of aggregate interferes with forming the proper kind of aggregate, which is what's involved in the case of CPEB. It's an aggregate you need. They've shown, at least in snails, that if you kill the CPEB, it can't learn. So I do think that this sort of focus on cytotoxicity as opposed to interfering with the memory mechanism may be one of the sort of elephants in the room of thinking about pathology here. But that's my personal view. Obviously, it's not a majority view.
Is there any computational strategy you could apply to target condensates with small molecules once you have a condensate model?
I think there's work by Robustelli where they have used atomistic simulations. So I think at the moment, as far as I know, it's only atomistic that you can do. So they've studied like small clusters of IDPs with a drug that was found experimentally by Xavi Salvatella in Barcelona to selectively partitioning condensates. And I think that's the only study I've heard about that, but I think it's a big goal of the field in general.
Okay, other comments on this?
I mean, this goes back to the previous question that if you need a stoichiometric inhibitor of weak interactions, this looks rather daunting to me, right? Because each interaction is weak. You have a rather large number of these interactions, and you would to shift the balance, you would need to inhibit a very significant fraction of them, presumably, to destabilize the droplet. So yeah, it's a, I think, fundamentally challenging problem. Not impossible, but challenging.
Yeah, we got a comment here from Mihai Mezei in New York on aggregation, that larger proteins have more flat surface, so it might help aggregation. I think this is kind of an answer or a comment related to both small molecule droplet formation as well as aggregation. Okay, I think we have probably one more question left. Which properties of molecular chaperone would be needed to act both as a suppressor of amyloid and LLPS formation of a client? I mean, this is a challenging thing, because I think that most experiments that at least I'm aware so far, they solve this problem by dissolving the condensate. Any other comments on this?
Yeah, but I think that would be the solution I would come up with as well. That is, if it binds to a form and in a way that is soluble, then both phase separation and aggregation could be prevented. But of course, that may not be that may only be a temporal way of slowing down the process.
I was
just
going to
because
make a
eventually.
comment on the previous question, which about the flat surface, which I thought actually was a very smart remark. I really enjoyed that remark. And it brings to mind this one feature that, normally, the proteins that, I used to work on and so on were all very small things. But you have to worry about the whole system, congealing on you. Whenever you have something that looks homopolymeric on the outside, then you better make sure it doesn't, attract itself. That's why it's very nice that DNA is all negative. It's very nice that actin is all negative. And so there's been a lot of, effort in evolution to make sure that those flat surfaces don't come together. Now, the problem is you want them to sometimes come together, and that's why you use regulatory proteins that are often intrinsically disordered and fuzzy. So this is I think the sort of thing here is that to control that ability to aggregate so that you just don't congeal into a mess all the time. I think that's the problem that has to be, that's sort of been solved in biology, but exactly physically how, there are probably many ways, but then you have to control the opposite thing. So I think we're seeing that the sort of skeletal regulation problem, the DNA regulation problem, like in the chromatin, are both very rich because of this. It's what makes them challenging and interesting.
Okay, thank you. So I don't see any more questions open. I think we had answered all of them. So I would like to thank primarily all the speakers for these wonderful talks, for the Protein Society to giving us opportunity to explore computational approaches to protein condensates. And well, thank you all of you for